Welcome, everyone. My pleasure to introduce Andre's dissertation defense.
Andre Moncrieff's dissertation.
Hey, Van. Yeah, make sure everyone has your microphone muted if you're online. So just a note to the committee that after a seminar, we'll adjourn for 10 minutes and then meet upstairs in Foster 214. Is that right, Andre?
Yes.
All right, thank you. At any rate, so Andre got his bachelor's degree, had actually two majors. He's a music major, so he got a BA. He's an amazing violinist. I'm sure you've all heard him play. And also got a BS in biology. He's one of the museum's birders from infancy. He's told me a lot of great stories about his family, nurturing that. I think they got stuck one time looking for some bird that he wanted to see. Anyway, it was very, very nice of them to nurture that love. I don't think any of the rest of them were birders, but at any rate. And as an undergrad, he got heavily involved in research. He worked on the Western glaucous wing gull hybridization complex out west. So that was a great opportunity. And I noticed that he had 3 publications by the time he finished his undergrad, which is amazing. All right, so he started the PhD program here in 2015 and landed one of those nice NSF graduate research fellowships. So he was supported for the first few years here by that. He's been extremely productive. He's got right now 12 publications, including 8 since starting at LSU. He's always been really good about publishing trip reports. So after an expedition, writing up a publication that summarizes the major findings of that. And that's great. Those publications probably outlast any of our other scientific things because they sort of lock in time what was found during those expeditions. And of course, we're in a changing world, so all that's going to change. He's either led or participated in 8 major collecting expeditions since being here. I credit him with really keeping our Peru expedition program going. He's been the one that wrote the permit request. He's involved in writing another one right now for our trips next summer. At any rate, so Andre's been able to blend his love of birds and field work to study what is really an iconic species, blue-crowned manakin. And so he's going to be sharing that research today. And if you're online, please mute your microphone.
Everyone, I'm just gonna get things started up here. All right, can people on Zoom still hear me? Thank you, Rob, for that introduction. And it's just really exciting to be here, seeing so many familiar faces and people on Zoom. Thank you for being here. A common thread through my whole dissertation is that rivers can act as barriers to the movements of different organisms. This river barrier effect has been known for a long time, and it's been especially studied in the Amazon basin of South America. The Amazon basin is one of the most species-rich areas on Earth, and you can see it light up here in this heat map of vertebrate species richness. And like a lot of other researchers, I'm interested in why this area of the world in particular has so many species and how they came to be. So one of the explanations for the high biodiversity in the Amazon basin is that rivers separate populations, which then evolve into new species. There's no other place on Earth with such an immense network of rivers. We've got the Amazon River running from its source in the Andes, east to the Atlantic Ocean, and then we've got many large tributaries that are generally angling east, flowing into the Amazon, and draining the water from the basin. Here's a photo of the famous meeting of the waters next to the city of Manaus in Brazil, where the dark Rio Negro meets the muddy Amazon River. And at the narrowest section that you see here where the rivers meet, the channel is still about 2.5 kilometers wide, which would fit over 7 Eiffel Towers laid on their side. And this kind of distance proves to be too much to cross on a regular basis for many birds, but also plants, primates, amphibians, many other types of organisms. And so we often see genetically distinct populations or related species that replace each other on opposite banks of these rivers. Another reason river barriers are often very strong for certain species is that these species can be restricted to what's known as terra firme forest. This is upland forest above the floodplain. And that can make a river like this one, the Juruá, which is just a few hundred meters wide, have an effective width that is much, much greater. In this case, 10 to 15 kilometers of floodplain forest separate quite a few species. Now, river width varies along the lengths of rivers. Where rivers begin, they are quite narrow. And in these headwater regions around the periphery of the Amazon basin, there's some data showing rivers are not much of a barrier. But these areas are also really hard to get to and sample, so understanding what's happening at the population genetic level around headwaters has been very challenging. And that's one of the knowledge gaps that I try to address in my dissertation. Another complex aspect to rivers is that the channels they flow through move over time. Here's a 30-year time lapse of a section of the Juruá River in Brazil, showing a process known as a meander loop cutoff that results in land getting passively transferred from one bank to the other. Meander loop cutoffs are a local type of river avulsion, which means a river channel shift. But larger river avulsions can also occur and passively transfer big areas that include terra firme forest. And this process has important implications for rivers as dispersal barriers. Again, we have very little empirical data on how these types of river changes affect the biogeography of organisms. Another knowledge gap that I try to address here. So the main thrust of my dissertation is to try and make a little more sense of how rivers in all their complexity influence the evolutionary process in birds, with special attention to what happens around river headwaters and river avulsions. I'll present my dissertation in 3 main parts. The first part is focused on the evolutionary relationships and taxonomy of birds in the genus Lepidothrix. To date, this is actually one of the better studied genera of birds in the neotropics, and I wanted to continue building on this study system to investigate genetic patterns around river barriers. And I see this part of my dissertation as really laying the groundwork for the next 2 parts. In part 2, I'll guide you through my work studying population genetic patterns around narrow headwater areas. And in part 3, I discuss how river avulsions can affect the evolution of populations, and I include a case study from some of my recent field work along the Ucayali River in Peru. To accomplish these goals, I relied heavily on research specimens rich with data, including high-quality genetic material, and I couldn't have asked for better support from LSU as I conducted collections-based field work. I also received specimen and tissue loans from many museums across the US and also from Brazil, so extremely grateful to those institutions. I prioritized visiting sites with very little inventory work, and these trips often involved camping for weeks at a time, long boat rides along remote rivers, carrying liquid nitrogen tanks over land, long distances. But despite all these challenges, it's been absolutely worth it. And this field work is what has allowed me to pursue the projects that I'm going to be talking about today. The genus Lepidothrix is a group of charismatic birds in Central and South America. Like other manakin species, the males, they get together in what's known as leks, and they display for females. They have many great dance moves, very acrobatic, and The males you can see here in this genus, they have brightly colored crowns that contrast with the rest of their plumage, also colors on their rump, and it's assumed that these help attract the females. So in order to better develop a study system for learning about biogeographic processes, I wanted to first get a better idea about the evolutionary relationships between the different members of this genus. Previous phylogenetic studies of the genus had involved just 1 or 2 individuals per species. So there's definitely some room for complementing previous studies. And there's a lot of geographic variation within some of these groups. So that was important to assess as well. Here are a few photos I took of blue crown manakins in Peru, showing some of the geographic variation. You can see birds with lighter crowns, darker crowns, greener plumage, darker plumage. And this was a goal of this part of the study, to get a better understanding of the relationships between these groups. So thanks to institutions across the US and Brazil, I assembled a set of tissues from 67 individuals across 17 of the 18 subspecies in the genus. And then I prepared RADCAP libraries for Illumina sequencing. And briefly, RADCAP is a method that combines some major benefits of a couple of common sequencing techniques. Like sequence capture of ultra-conserved elements, RADCAP results in high coverage of target loci, including degraded DNA samples from historical specimens. In my work, I sequenced DNA from toe pads of birds dating back to the year 1958. And this really helped me complete my geographic sampling, although you can never really be complete. So RADCAP, it also shares benefit of RADseq in being relatively cost effective. So this is helpful when you're sequencing large numbers of individuals. I used several different tree estimation techniques that produced highly concordant results. So here's one of the trees I built using the program IQ-Tree based on over 5,000 SNPs. Several species within the genus replace each other across rivers, but one of the most important contributions of this study was to clarify relationships across the Andes, where there are 2 populations within a group, the blue crown manakins. So in the dark orange, you can see populations in Central America and the west coast of Colombia and Ecuador. And this group formed a monophyletic clade. There are also phenotypically variable populations of blue crown manakins throughout the western Amazon basin, shown in light orange, and this group also formed a clade. The presence of these 2 divergent clades brings up a frequent issue in taxonomy, which is how do we decide if we have 1 or 2 species when similar populations are allopatric? Ideally, we'd be able to assess directly whether these populations can interbreed or not, but we can't. However, different vocalizations in birds can be a really good indicator of reproductive barriers, especially in birds like manakins, which are suboscines. They have genetically hardwired songs. And in manakins, both the calls and the songs are a prevalent part of their lek displays, so they're very likely to function as prezygotic barriers. So let me just walk you through how voice could help with a taxonomic decision. So first, how different are the vocalizations between these 2 groups? We can visualize and quantify vocalizations by looking at spectrograms. And for simplicity, we'll just look at the calls of these birds. In Central America, they have a rapid trilled call, but in Amazonia, populations give a sweet rising whistle. So the vocalizations are completely different. You never hear Central American birds giving the Amazonian call or vice versa. Now, a follow-up question would be, how different are the vocalizations of any co-occurring sympatric species of Lepidothrix? And it turns out that blue crown manakins and cerulean capped manakins are sympatric right at the lowest foothills of the Andes. So how do their vocalizations compare to each other? When we look at their calls, these sympatric taxa have very similar rising whistles. And when I listen to them, I can't even tell them apart. We can use this finding as sort of benchmark to say that vocalizations of blue crown manakin populations in Central America versus Amazonia are more different than necessary for maintaining reproductive isolation. With this vocal comparison completed and additional support from genetic and phenotypic data, I made a recommendation to elevate both of these groups to species level taxa, and they're now recognized as the velvety manakin and blue-capped manakin. I actually submitted a proposal to the South American Classification Committee earlier this year, and that was passed in July. So now there's broadly recognized two species level taxa here. So one of the end results of this phylogenetic study is what I consider to be a better estimate of species level diversity than we had before. And this is using a pretty conservative biological species concept approach. The reality is that these types of taxonomic splits are occurring all the time. When I started birding back in the year 2000, there were about 9,700 species of birds recognized. 22 years later, we're at 10,900. This is well over a 10% increase. And our goal as systematists is to really describe as accurately as possible the diversity that exists. And at the species level, this is very important in terms of conservation, but also for research in biogeography, macroevolution, and other fields that benefit from good taxonomic work. So to conclude, in the first part of my dissertation, I conducted a phylogenetic study of Lepidothrix manakins. This helped clarify relationships within the genus. I found that the blue-capped manakins of the Western Amazon Basin make up a clade that deserved recognition as a distinct species. And with this knowledge, I think I'm in a better position to carry out a more detailed population-level study of blue-capped manakins. And this is what I do next. So in part 2, I used blue-capped manakins as my study taxon to explore population genetic patterns around river headwaters. And one potential implication of these headwaters is that by allowing for more dispersal, rivers may not be sufficiently strong barriers to account for allopatric speciation. So the presence of headwater regions allowing for dispersal has often been used as a critique of the riverine barrier hypothesis. This hypothesis goes back to observations by Alfred Russel Wallace, before him to indigenous people of the Amazon Basin. In 1852, Wallace said in reference to primate species that the Amazon, the Rio Negro, and the Madeira form the limits beyond which certain species never passed. The native hunters are perfectly acquainted with this fact. And he went on to say that on approaching the sources of the rivers, they cease to be a boundary. Now, these statements from Wallace are about species distributions. They don't explicitly propose a role for rivers in allopatric speciation. And this would come later with researchers like Helmut Sieck. And he proposed that populations of birds in the Amazon region originally lived in a continuous forested habitat, which was then dissected into many interfluves during the formation of the Amazon River drainage, causing allopatric speciation and the frequent opposite bank species replacements that we see today. And this has been referred to as the basic riverine barrier hypothesis. This basic model has several problems, including current data suggesting that the Amazon drainage was established by the late Miocene, which is older than a lot of species level diversity found in Amazonia. So if rivers are to account for speciation in the Amazon, there needs to be some level of dispersal across rivers to account for younger species. Here are just a few of the many species pairs separated by the Negro River. And one way to account for their variability and divergence times is by different dispersal histories around rivers. And one way that birds can potentially disperse to the other side of a large river is by crossing the narrow headwaters. I've circled some of the main headwater regions within the range of blue-capped manakins, where I suspected I might find populations with greater genetic connectivity. Most studies looking at headwater biogeography have been limited to studying patterns of species distributions. But for my dissertation, I was interested in taking a finer scale view to see if within an Amazonian species, there's consistently greater gene flow around headwaters. Now, blue-capped manakins are a popular study taxon. In the 1980s, Angelo Caparella, who got his doctorate here at LSU, collected a legacy series of these manakins around the Amazon and Napo rivers to conduct the first genetic tests of a river barrier effect in birds. He was able to show that the Amazon and the Napo separated distinct genetic clusters, even though the birds in this area look very similar. And this brought up the exciting idea that there could be cryptic diversity across a lot of barriers, and we now know that this is the case. So thanks to Caparella's targeted work and general collections from many other institutions, the blue-capped manakin is one of the best sampled Amazonian birds in natural history collections. And it's also a nice species to study when looking at riverine barriers because it's quite closely tied to terra firme forest. Early on in my doctoral work, I collected a lot of data on blue-capped manakin plumage coloration using a spectrophotometer, looked at a lot of specimens from different museums in the US and Brazil. And I found some interesting patterns of phenotypic variability. You can see the distribution of the manakins is colored by their approximate body plumage color. And particularly, I noticed around the headwaters of the Ucayali River, there was a lot of phenotypic variability. And at this early stage of the dissertation, I hypothesized maybe this variability around the Ucayali headwaters is due to gene flow across the headwaters, a river headwater effect. To really test this hypothesis, though, I would need to get a lot more samples from this region. So I led a targeted expedition for 48 days in 2018, in which we boated up and down the Ucayali headwaters. This was to some of the most remote places that I've been in the Amazon. At one point, we were 18 hours by boat from the nearest road accessible town. And we collected 80 specimens of blue-capped manakins. This is really an unforgettable experience. Then came the lab work. I received great help. There's only one slide dedicated to this, but not proportional to the time spent. I received great help from Rosalind Remsen, who worked with me for 2 years in the lab as an undergraduate student to prepare genomic libraries. We ended up with a genomic data set of 706 individuals from 83 localities across the Amazon basin. The sampling also included 13 degraded or historical DNA samples to fill out sampling in several regions. So now let's look at some of the results. I first want to take a few minutes to appreciate the broad picture. And one helpful way to visualize gene flow across the landscape is with a method known as an estimated effective migration surface. This is a method based on a matrix of genetic distances among georeferenced individuals. And it takes into account isolation by distance and visually highlights areas where genetic distances are higher or lower than expected according to the model. So especially low migration areas are shown in the dark red tones. With my data here, I was pretty excited to see that dark red, low migration areas correspond to river barriers, the Amazon and the Ucayali River in particular. And this method is agnostic to the presence of rivers. Another helpful way to look at the results is with a principal components analysis of the SNP data. And the PCA shows at a broad level, just kind of eyeballing it here, that there are 3 main genetic groups. The first group corresponds to individuals north of the Amazon River. A second group corresponds to individuals in the Andean foothills west of the Ucayali River. And the PCA shows a classic isolation by distance pattern here with individuals strung out along a north to south axis. This makes sense given the substantial topography of the Andean foothills that would lead to lower population connectivity. And the third group covers a very large area, but it's quite closely clustered. And this group corresponds to south of the Amazon and east of the Ucayali River. Also point out an intriguing intermediate sample from the south bank of the Marañón River, which is a headwater of the Amazon. I really wish that I had better sampling here. This is just one individual, but it's very suggestive of a headwater effect around this headwater, the Marañón River. And more broadly, I've circled a swath of the Amazon basin along headwaters from northern Peru to southern Colombia. And this is from the Marañón River to the Caquetá River. I was interested to see a lack of the dark red tones that denote lower effective migration. I interpret this as some evidence for greater genetic connectivity around these headwaters compared to downstream areas. However, when we look at the Ucayali River headwaters, which is about as far up the Amazon drainage as you can go within the range of blue-capped manakins, we do see those dark red tones that denote lower effective migration. This is not the result I was expecting. To explore these genetic patterns further, I ran a program called SNMF, and this program estimates ancestry coefficients for all individuals in different numbers of populations. And it turns out that 5 or 6 populations adequately describe the genetic variation. And I've displayed the results for 5 populations with averaged ancestry coefficients for each locality shown as pie charts. Again, a big takeaway here is that the major genetic transitions in the Amazon basin occur across river barriers. To emphasize this point, the largely pink pie charts show that birds from the south bank of the Amazon and east of the Ucayali are genetically clustered with birds all the way south to central Bolivia, and not with birds a few kilometers away on the other side of the Amazon. We can also zoom in on the localities that Angelo Caparella visited in the 1980s. And similarly, I found distinct genetic breaks across the Napo and the Amazon rivers. Here are some localities on opposite banks of the Caquetá River in Colombia. And interestingly, here we find far more similar ancestry coefficients than at localities across lower parts of this river. And that's what I would expect given a river headwater effect. But now I want to zoom in on the headwater area that I've invested most in studying, and that's the upper Ucayali, where I had hypothesized headwater hybridization was occurring. To my surprise, the genetic results showed very distinct clusters on either side of this headwater. Now let's look at the phenotypic data too. Here I'm showing the ventral and dorsal side of specimens I collected in 2018. And east of the Ucayali and the Urubamba, there's a cline which is shown by the orange arrow there, where dark individuals in the north transition to this kind of dark green color. This transect is about 180 kilometers. So it's a gradual change from black to green. But on the west bank, there's a noticeable jump to an even brighter green. These birds have bright yellow bellies and sky blue crowns. Now, perhaps you're wondering, what about sampling between the Tambo and the Urubamba rivers? That was a big question of mine as well. So let's look at the landscape in a little more detail. Here's the satellite view, and we have the Tambo and Urubamba rivers flowing north and joining to form the Ucayali, which is about 600 meters to 1 kilometer wide at this point. And one of the first features of the landscape that pops out is the Tambo and the Urubamba are both hugging the Andean foothills. And this is right where those brightest green individuals are found. On the other side of the headwater, we've got the dark black plumage and the dark green. These birds are separated by usually a few kilometers of floodplain forest. And the genetic break is very distinct. In between this area, though, despite my very best efforts over 9 field days, including long hikes into good quality terra firme forest, doing playback for these birds, usually I'd find them within a day. I could not find any individuals of blue-capped manakins. I would hesitate to say that these birds are completely absent from the area, but it seems that at least it could be a low density area. So why isn't there hybridization across these headwaters? Well, in this case, I speculate that there could be a preference for foothill forest in the birds west of the Ucayali and Tambo rivers. This may be enough to isolate the populations. There could also be a real distributional gap between these populations. And perhaps if dispersal did occur and bring these forms into direct contact, the plumage differences would be enough that these populations would mate assortatively. So I'd definitely like to do more sampling in the area. And there are a number of other taxa that replace each other across the Ucayali, which would be interesting to explore. Now that we've had a more in-depth look at the region, it appears that there's a transition from black to green forms that I documented on the east side of the headwaters that involves hybridization strictly on that side of the headwater. And this appears to just be a transect through a contact zone that extends about 1,500 kilometers east all the way to the Madera River between these two forms, not involving the brightest green forms at all. So in conclusion, I found evidence that Amazonian rivers structure genetic variation in blue-capped manakins. The Amazon and the Ucayali rivers appear to be particularly important in this case. I found evidence for increased gene flow across some headwaters, like those from northern Peru to southern Colombia, which supports the hypothesis that dispersal around headwaters serves to connect populations and limit genetic divergence across rivers. But in contrast to my expectations, the headwaters of the Ucayali River separated distinct populations. In this area, the habitats differ on each side of the headwater, so it's difficult to distinguish between riverine or ecological barriers that may be preventing gene flow. So I consider this to be an unusual headwater and perhaps an exception to a general pattern. A small distributional gap and phenotypic differences between populations may also be preventing gene flow in this area. So in this section, I've shown some evidence for dispersal around headwaters, but it's possible that dispersal could be happening around wide areas of rivers as well. And one of the possible mechanisms for this, beyond just a bird deciding to fly over a river, is passive transfer via river avulsions. This is what I want to focus on in the final part of this talk. Research on the relationship between river avulsions and Amazonian biogeography is still very much in the early stages, but I think there's a lot of potential here for developing our understanding of the origin of Amazonian biodiversity. First, I'll synthesize some information relating river dynamics to Amazonian biogeography. This forms one of my dissertation chapters that I'm working to publish as a perspective piece. And second, I'll be sharing a case study of the biogeography around a specific avulsion area that I visited in Peru. And this study was published previously in the Wilson Journal of Ornithology. The idea that rivers in the Amazon basin have had a long and complex history of movement has been around for some time. So here's some images derived from airplane radar that were used to challenge the biogeographic tradition at the time, which was really focused on habitat shifts during the Pleistocene. And in the words of Rasanen and colleagues, they found evidence for long-term fluvial perturbance, or in other words, signs of shifting river channels over time. Now, images from aircraft and satellites have been really key in helping us learn more about these processes. And these images just keep getting better and better with time. For instance, here's some recent Landsat imagery of a complex area along the Amazon River in Brazil. We have accessible satellite images like this one going back to the 1970s and 80s, but they've definitely improved. And we also have a great elevational model from the International Shuttle Radar Topography mission, which flew in the year 2000. And This data makes it a lot easier to see landscape features like paleochannels, which are areas where rivers used to flow. So in this SRTM image, the dark areas correspond to bodies of water. The medium gray is floodplain, and those lightest patches correspond to higher elevation terra firme forest. And someday, very soon, I really hope to be able to sample birds in that patch that I've encircled there. It's a strip of terra firme forest, which is over 50 kilometers long. It was identified in a recent study, although no biologists have visited there as far as I know. The current Amazon River channel flows south of that landmass. And at some point within the last 50,000 years or so, the Amazon flowed north of that landmass. Now, in biogeographic studies, it's often very difficult to confidently link a specific datable landscape process with generating allopatry of populations. But I think that river avulsions offer a fantastic opportunity to do just that. So if large avulsion events like this one can transfer hundreds or thousands of individuals of a species across a river on a semi-regular basis over evolutionary timeframes, then that could potentially be a mechanism for increased speciation rates. So do avulsion processes help explain the origins of Amazonian biodiversity? This question has received quite a bit of attention, but almost exclusively from the perspective of aquatic organisms, particularly fish. So kudos to the ichthyologists for leading the way on this. Most work is focused on a type of avulsion known as river capture. To give an example, here are two river drainages, the top drainage flowing north and the bottom drainage flowing south, each with their own communities of aquatic organisms. Then in the avulsion process known as river capture, the top drainage can capture part of the bottom drainage. This means that any fish or other aquatic organisms are then separated from other populations in the bottom drainage. This process can happen again, with the bottom drainage taking on a good chunk of the top drainage, along with any aquatic organisms in the captured rivers. It turns out that these processes are very important in explaining diversification of fishes in the Amazon basin and elsewhere, and it's provided substantial support for the river capture hypothesis. This hypothesis states that river captures promote dispersal and allopatry and eventually speciation within river channels. These types of events have many different causes, pretty much anything that causes a lot of erosions. These can be flooding events, earthquakes, and more. So although to date, the focus of river capture biogeography has been focused on aquatic organisms, these captures can also alter the distributions of the organisms found around these rivers as well, the terrestrial taxa. And also for terrestrial taxa, an avulsion involving a single river channel can also promote dispersal and allopatry. That doesn't need to be a capture event of other rivers. For instance, here are two terrestrial species separated by a river and its floodplain. And in this simplistic model, a river avulsion could passively transfer species A to the right bank, where several different possibilities could come to pass, including extinction, diversification, or introgression. One of the few studies to look for evidence of gene flow around avulsion sites was conducted by our own LSU researchers. And in this case, they were looking specifically around meander loop cutoffs of the Mississippi River. And they found evidence that these cutoffs promoted gene flow between skink populations across the river. And I suspect that this sort of phenomenon is happening regularly in the Amazon basin, but we're really just scratching the surface in adequate data to study this topic. Most of the evidence that we have regarding the importance of river avulsions is indirect. For instance, the variable divergence times across rivers suggests dispersal is happening, and so avulsions might be contributing to that process. There's also evidence of past introgression between a number of opposite bank taxon replacements across the Madero River, which is thought to have shifted substantially over the last 100,000 years or so. But in order to confidently link specific landscape events with biogeographic outcomes, seems to me that looking at relatively small, well-defined and dated avulsions is a great place to start to complement broad-scale phylogeographic research. So with these ideas in mind, I led an expedition in 2019 to the site of a river avulsion on the Ucayali River in central Peru. You can see from this satellite image that it's a very complex network of rivers and lakes in this area. The largest river here is the Ucayali. It's about 600 meters to a kilometer wide here, and it flows north. And geological data, historical accounts and maps, they document a rapid shift in the course of this river about 230 years ago, or around the year 1790. And its present course is shown here. If I overlay the old and new river courses, we can see that there's a large area in the middle, about 2,000 square kilometers, that at different times has been on the west side of the river or on the east side. And I'll call this area the avulsion landmass. One of the clear remnants of the avulsion is that big blocked valley lake in the middle of the landmass. And then there's also some evidence of a paleo channel along one side. Now we have pretty good sampling at localities outside of the avulsion landmass, and we know that there are about 17 closely related taxa of birds that replace each other across this part of the Ucayali River. These birds include jacamars, puffbirds, and of course, blue-crowned or blue-capped manakins now, which have a green subspecies and a black subspecies replacing each other across the river. The big question that I have is, what do we find within the avulsion landmass? My favorite hypothesis before doing field work in the area was that given how recent this avulsion is, we should find some populations of West Bank taxa in the avulsion landmass. And possibly we could even demonstrate some hybridization between these river delimited taxa, which are often very closely related. So just to visualize the possible outcomes, we might find that populations of the West Bank taxon have held out the past couple hundred years in this landmass. We could find East Bank birds have recently colonized the area, perhaps outcompeting their close relatives. We might find evidence of hybridization or maybe coexistence of the two forms in sympatry. So what did we find? We were able to collect specimens of one of the members of nearly all of the taxon pairs that replace each other across the Ucayali River. And in the vast majority of cases, we found that the East Bank taxon occurred in the avulsion landmass, suggesting recent colonization by these species. In two cases involving Pionides parrots and Fugopedius wrens, we found hybrids within the avulsion landmass, but we also found evidence of hybrids in other areas on both sides of the river, suggesting it's not just this avulsion event, but a longer history of crossing the rivers that gives that pattern. And then very tentatively, it looks like two West Bank taxa had populations lingering in the avulsion landmass, although the identification of these particular taxa is quite difficult, even though we have some comparative material here, and I'd really like to get genetic data on these birds. Looking at our blue-capped manakin specimens, you can see that the taxon from the avulsion site morphologically matches the East Bank taxon, which is not what I had hypothesized. And again, it suggests that recent colonization of the avulsion landmass. This result gives empirical support to the idea that even a relatively large river avulsion doesn't necessarily have a lasting impact on geographic distributions or patterns of gene flow for river delimited taxa. For the blue-capped manakins, I also obtained genomic data that further indicates an absence of hybridization across the Ucayali. And for me, this raises the question of just how big or recent does an avulsion have to be to leave a clear biogeographic impact on the region. One of my upcoming projects will be to expand this genomic data set to a lot more taxon pairs across the Ucayali River to further explore these patterns. In part 3, I've explained how river avulsions provide a mechanism for dispersal and subsequent allopatry of terrestrial taxa. But biological sampling is extremely sparse around well-defined avulsions, and more work is needed to link these specific landscape processes with biogeographic outcomes. I also found that the biogeographic signal from a very recent avulsion along the Ucayali River appeared to rapidly decay, although further genetic work is still pending. To review the arc then of this dissertation, I first conducted a phylogenetic study of manakins in the genus Lepidothrix that clarified the taxonomy of the group and set the stage for the population level studies that I would conduct next. In part 2, I found some evidence around river headwaters for greater genetic connectivity, which softens the river barrier effect and suggests that other barriers in addition to rivers may be important for generating allopatry and speciation in some cases. And in part 3, I discussed how river avulsions occur regularly at multiple scales in Amazonia, and that they could potentially promote dispersal, allopatry, and speciation. I also found that a recent river avulsion in Peru appeared to have a limited effect on the biogeography of birds in the area. So one of my next sampling priorities is to visit a slightly larger avulsion. In this dissertation then, I've tried to bring us a little bit closer to understanding how rivers in the Amazon basin have shaped the evolution of bird populations. I think obtaining more biological samples, both from avulsion areas and from headwater areas, will be key going forward. And also collaboration with geologists will be important. I think getting more sediment cores where we can date specific landscape features will be important for constructing the chronology of the Amazon basin at a finer scale. And this work is already underway, and I'm excited for what that will bring. Many thanks to so many people and institutions that have helped this work be possible. None of my work in Peru would have happened without the support from collaborators at Corbidi and research permits from CERFOR, also the many funding agencies that gave me the flexibility to pursue these projects. I also received so much help from indigenous communities in Peru over the years, and I feel super lucky to have overlapped with so many talented field biologists that I get to call my friends and collaborators. Thank you so much to the many important people in my life who have supported me, guided me through this journey at LSU, and thank you to my undergraduate mentors for inspiring me to pursue a career in biology. Lastly, to my family, you've always been there for me. Thank you so much. And I'd be glad to take any questions. Yeah, Kyle.
I wonder about the habitat in that area in chapter 2. I guess it's between the Tambo and Urubamba.
Urubamba, yeah.
Urubamba. Yep. So it looked pretty flat in the satellite image. Could it just be a really big floodplain?
So you're talking about in between those two rivers. Yeah. There is, yeah. So I mentioned that there were 3 sites that we visited within that area. One of the sites I had to hike almost 10 kilometers to get to a terra firme plateau, but most of the area is about 300 meters in elevation. I was able to gain about 150 meters as I did that hike. But then we camped one night, surveyed that area, and had to hike back. So that was probably my best shot at finding a population of these manakins, which I wasn't able to do. But I think it's still possible they could be there. Yeah. Mike.
I had 2 questions that kind of relate to female choice and mating preferences. So one was, you mentioned in Sabasins that the male vocalizations are genetically program? Do you know anything about the genetic basis of female receptivity or preferences for those balls?
I don't. Genetic. I'm not sure if that's really something that's been studied. Yeah.
And then another one sort of bounces off Kyle's question, then, that we know that a lot of mating signals that are, the visual or home are context specific. And so that the same signal presented in say light environment could be perceived differently by a potential mate. So again, looking at that break where you up between the flatter forest and the more mountainous foothills of the Andes, are the, as you perceive it, are the light environments in which the manakins are displaying in these places radically different or noticeably different, or potentially are the oral environments just because of just the density of trees or what have you? Did they start to be different?
That's a really good question. For the people in Zoom, I'll just repeat that. Forgot to do that for the last question. But basically, are the light environments potentially different between areas where different populations of the blue-cap manakins are found? So one of the populations, the one in the foothills that I found, it occurs within like a stone's throw of the river. And for me to say that habitat right at the very base of the foothills is different than the habitat on the other side, I'm not sure I could say that. And I did find a little bit of variability in kind of how thick the vegetation was, depending on the different lek that I found. So to associate that with the different populations, I'm hesitant to say that I could. But yeah, that's a really interesting question. Jackson.
Yeah, for your part two, so your sample size, you only did 706 individuals.
Correct. Yeah.
Just wanted to make sure I got that. But no, my real question is, for part two is regarding terra firme. So in relation to the river, is that type of habitat, do you expect the same abundance of manakins, whether that is, if that terra firme is located adjacent to headwaters versus the much larger channels? Like, is that habitat, once you forget the rivers, is it identical? Or could there be like micro effects of proximity to that large body of water affecting abundance, or not just specifically gene flow or lack thereof?
So you're talking about the difference in terra firme habitats between headwater areas and lower areas?
Yeah. Or if if someone dropped you in from a helicopter, like blindly, could you tell if you were near the headwaters versus the large?
I wish I was a better botanist. I don't think I would be able to tell. I think most of the differences that we see in habitats around the rivers depends on specifically what river you're looking at. So the Judois River, for instance, that I showed early on, with all those meanders, really wide floodplain, you have to be much further from the river to get to the terra firme. And other rivers, it's like right on the bank, just because the river kind of incises more deeply into the terrain. But yeah, I think it would require a specialist to be able to look at the plants, to be able to comment on that. Yeah, thanks.
So I'm guessing these river erosion events, which Jackson and I talked about at length, are not random, right? And so is it possible that these events are the result of, heavy years of rain that cause massive flooding that cause the river changes, and that makes the terra firme land basically uninhabitable for the birds, and so it's basically this dead zone for a decade or more that is then recolonized from the birds on that side of the river.
Yeah. So Chris just raised a good point, which is it possible that during these avulsion events, there are large flooding events? And that could potentially just wipe out populations in the area. So we might expect new colonization after that event is over. And I think that very well could explain the result that we saw in the Ucayali. The area in the avulsion landmass is what I would call near terra firme. It's not quite as high as terra firme in other areas. In our tags, we wrote transitional forest. Even though we had quite a few terra firme birds, like the blue-capped manakins, one possibility is they were just wiped out. I think there are some areas like the one avulsion that I highlighted where it's probably just too high, the terra firme there, to get flooded out. But there is evidence also, like you're saying, these are not just random events. The Western Amazon Basin, which is lower lying, has more frequent river avulsions than areas on the Guiana Shield and Brazilian Shield, where the rivers flow a lot straighter. So I think the explanatory power of these river avulsions is limited more to the western half of the Amazon Basin.
You have questions? All right. Thank you.
All right. Thank you. Thank you so much.